Woolcock, and I go to the Academy of Charter Schools. I'm excited to share my science project with you. My project explores how we can simulate and address cellular damage in onion epidermal cells, representing damage similar to that seen in human skin cells affected by progeria. To start my experiment, I take an apple and cut it very smallly into thin strips. Then I put it through a strainer multiple times and use a spatula to gently push it down to try and get that juice from the apples. Then I cut up the grapes and do the same process, cutting them up and putting them through the strainer very carefully. I use rubber gloves to avoid losing any remedy and to try and not get it on my hands. Also, I switched to a bigger strainer so that you could get more juice inside the apples. After putting it through the straining process a couple times, I realized that it is easier to crush the apples using a whisk than putting them through the strainer. Lastly, I take a plastic container and try and avoid any air or anything else getting into it to try and make sure that the remedy is safe and secure. I extract it into the remedy and then make sure there's nothing else in it. Now, inside the container, I add a few vitamin D capsules, which I gently have to cut open with using a knife and extract the inside of the vitamin. I do that 3 times and then use my whisk and mix it all together carefully. In my experiment, I used 3 types of control groups for the treatment. First, I apply the hydrogen peroxide to simulate damaged cells. This represents the cells with oxidative stress, which could technically represent Hutchinson-Gilford Progeria Syndrome. This is a syndrome where people at young ages can age really, really fast and eventually leads to death. I use this process to simulate the certain skin cells in progeria. For the second control group, I applied hydrogen peroxide to simulate the progeria. Then on top of that, I add the remedy. Using a pipette and slides, I take my carefully cut up onion and I peel it off of the layer of the white to get to the epidermis. I use the remedy with no hydrogen peroxide first, and I gently put it under the microscope. And as you can see, the cells look fine. They look healthy and basically like normal human skin. Then I take my second slide, which this is the protein one, and you can see how the cells are pretty damaged and how using just hydrogen peroxide makes them kind of split apart and makes that small thin line much more blurry and not able to see it as well. That is representing the damage from progeria. But once I made a third slide, I realized after putting hydrogen peroxide and then letting it sit for a little bit and then putting my remedy on top of it, it was much less blurry and it was much easier to recover almost all of the line, and it was not nearly as blurry as it was before. And I was really able to see how using the remedy, it can help bring some of those cells back to life and potentially could cure it altogether. So after looking at that third one under the microscope, you could see how it's much more clear and just overall easier to see. And it definitely helped show how the combination of vitamin D helped protect the cells from this stress and this might end up being a promising treatment for skin conditions similar to progeria. Using this method, it can also likely help other skin diseases or just rapid aging similar to child progeria in general or adult progeria. Using the 3 different slides, I was able to find that regular human skin is a lot more hydrated and it doesn't have as many wrinkles on the surface layer, and that's what the epidermis of the onion was representing, the surface layer of the skin. And then I was able to realize that the remedy can go deeper into your skin. So with people who have skin conditions like progeria, it could potentially cure it altogether. Thank you so much for watching, and I hope you found my project interesting. This research could inspire future studies on natural remedies for skin cell protection. Thank you so much. Have a good day.